All right. So it's 1 o'clock. We'll go ahead and get started. You can see that the poll has been launched. So if you want to go ahead and answer that as we go through the introductions. I'm Jen Kiblinger. I'm the scientific director at Dia Pharma. My colleague, Dr. Olivia Stricker, our business development manager, is on as well. We will be moderating today's webinar on cytokeratin 18 in MASH drug trials. Dia Pharma offers the M30 apoptosense and the M65 ELISAs in North America from our partner BLV Bio. Note that BLV Bio products are for research use only in the US and Canada, not for use in diagnostic procedures. It's my pleasure to introduce Dr. Jessica Toohey, our Senior Scientific Liaison for Dia Pharma's biomarker portfolio, which includes assays for studying liver injury. Jessica received her PhD in pharmacology from the University of Colorado Health Sciences Center, where she trained in mass spectrometry of lipids. She contributed some of the early work in lipidomics in 2 research positions, first at Cell Therapeutics in Seattle and at Vanderbilt University in Nashville as part of the Lipid Maps Consortium. Dr. Toohey has prepared a 30-minute talk on cytokeratin 18 as a biomarker in MASH studies, which will be followed by an opportunity for questions. So please feel free to enter those questions in the Q&A section throughout the presentation. And I will now turn it over to Jessica.
Awesome. Thank you so much for the introduction. All right, I'm going to end the poll. Thanks for everyone that answered. Looks like a lot of you are already familiar with CK18, and some of you are also using CK18 in your trials. But hopefully, I can add to your current knowledge and understanding. So I am going to share my screen. See how I do this. Oh, a second. So let me have you do this because for some reason there we go. Share screen. Sorry, it's always these technical issues that I am most afraid of. All right, okay. So I hope that everyone can see my slides. All right, so thank you everyone for joining. Thanks for giving me your time. I am really excited to talk about 2 of my favorite topics, which is CK18 and MASH clinical trials. As Jen said, it's going to be about 30 minutes. All right, let's get started. So the topics I'm going to cover today is I'm going to give a little bit of background of MASLD and MASH. It's probably going to be review for most of you. I'm going to talk about MASH clinical trials in general, and then specific endpoints associated with MASH clinical trials. I'm going to give an introduction to CK18 and talk about data from MASH clinical trials that have used CK18 as a biomarker. And then I'm going to finish up with some data of CK18 specifically related to liver histology. And then there should be plenty of time at the end for some Q&A. All right, so first things first, I'm sure most of you are already aware, but about a year ago, the new nomenclature for steatotic liver disease was released, and it's been adapted by multinational societies, including ASLD, EASL, and ALE. So the new term for fatty liver disease is now steatotic liver disease. MASLD is now metabolic dysfunction associated steatotic liver disease, a mouthful. So it's MASLD or MASLD. NASH is now MASH. And in addition, there is now a new category and a new term that is MET-ALD, and that describes the occurrence of MASLD with significant alcohol consumption. And I'll try to be very mindful in this webinar to use the correct terminology, but I'm nervous, so I may slip up, so please forgive me. And then also some of the slides and graphics that I'm presenting may still have the old terminology, so just want to make sure that I'm clearing up any confusion in that regard. All right, I could talk about MASH all day long, but I'm going to try to keep this to a half an hour. So I'm going to go over the next few slides pretty quickly, since it's going to be more review for most of you. What is MASLD? What is MASH? MASLD is an umbrella term that refers to a spectrum of conditions where there's accumulation of excess fat in the liver of people who drink little or no alcohol. It's a progressive disease that starts with relatively benign accumulation of fat in the liver, steatosis, but then patients can progress to MASH and ultimately to cirrhosis and liver cancer. Over the past 20 years, the incidence of MASLD has more than doubled, and it is now the most common chronic liver disease in developed countries. In 2021, the worldwide disease prevalence was about 38%, and that number has been forecasted to increase to 55% by the year 2040. So at that point, over half of the global adult population will have MASLD. So needless to say, this disease poses a significant health burden, and that burden is only increasing. The increase in prevalence is strongly connected to the epidemic increase in obesity and type 2 diabetes. MASLD is a complex and multifactorial disease. MASH progression results from numerous events originating within the liver, as well as signals from the adipose tissue and the GI tract. And one thing to point out is that MASLD is generally a very slowly progressing disease, so it can take years or even decades to progress from steatosis to cirrhosis. And I'll come back to that point in just a couple more slides. Until very recently, there were no FDA-approved drugs specifically for the treatment of MASH. So up to that point, really, the only treatment options were lifestyle changes, so improved diet and exercise, maybe bariatric surgery, and then therapeutics for comorbidities, so anti-diabetic drugs and lipid-lowering agents. But again, these are not specific for MASH. On March 14th, the FDA approved resmetirom, or Rezdiffra, for the treatment of MASH. And that was truly a historic milestone, and a huge congratulations to everyone that was involved in this effort. This approval will have a really big impact, not just on patients and providers, but really on everyone in the field, and also in regard to MASH drug development. As I just mentioned, MASH is a multifactorial disease, and different patients will likely have different underlying etiologies. So one drug or the same drug may not be equally effective in every patient. And there are many more MASH drug candidates that are in development. And the complex etiology of the disease offers many therapeutic targets. And the number of MASH trials that are initiated each year has been increasing exponentially. When I updated the slide the last time, which was in 2020, there were only about 80 MASH trials underway, and now it's over 120. So that's a 50% increase in just 4 years, and that's despite any slowdowns that were caused by the pandemic. So again, many more drugs that are in development, and even more drug candidates that are entering into pipelines every day. Again, not going into too much detail about this, since you're probably all very familiar, but because MASLD is such a slowly progressing disease, you can't really use hard outcomes as endpoints for drug approval, because you would need a ton of patients that would need to be followed over many years, and that's not feasible even just from a financial perspective or standpoint. So instead, the FDA and EMA are accepting changes in liver histology as surrogate endpoints for accelerated or conditional approval of MASH drugs. Treatment responses are determined by the evaluation of liver biopsy samples by a pathologist, and changes are measured by the NAFLD activity score, or the NAS, which is a composite score that's made up of the extent of steatosis, lobular inflammation and ballooning, and the NAFLD fibrosis score. So one analogy that I've heard to relate the NAS and fibrosis, and that I've always found super helpful, so I wanted to share this, is that of a road trip. So the fibrosis stage will tell you how far you've gotten along your journey, so where you are in the disease, and the NAS tells you how fast you're progressing in your journey. So again, I've always found this really helpful, so I just wanted to share that. But going back to endpoints, the surrogate endpoints that are accepted by the FDA for MASH drug approval are either the resolution of MASH without worsening of fibrosis, an improvement in fibrosis without the worsening of MASH, or both. And for EMA approval, both 1 and 2 need to be met. Now, the histological surrogate endpoints that I just talked about require a liver biopsy sample. And we could belabor all of the drawbacks and issues that are associated with that current requirement of biopsies and changes in histology. But what's most important, especially for early phase trials, is that it's really not feasible to use serial biopsies to assess drug treatment effectiveness or efficacy. And that's mainly because significant changes in liver histology may take a long time to manifest, or at least longer than the duration of shorter early phase trials. And for sure, the advent of AI-assisted digital pathology has really revolutionized liver pathology. And I'm always impressed by how granular and how sensitive these technologies are at detecting changes, at least as compared to traditional histopathology. But they still require a liver biopsy sample. So there's been a ton of interest in identifying non-invasive biomarkers that can not only detect treatment effects at early time points, but that can also give an indication of changes in histology later on. Imaging tools like ultrasound, MRI, MRE, PDFF are incredibly useful for assessing steatosis and fibrosis. And specific serum biomarkers, as well as panels of biomarkers, lipidomics, metabolomics, proteomics, have been used to monitor changes in other disease parameters such as inflammation, oxidative stress, and apoptosis. And the advantage of these non-invasive tests is that they can be done repetitively and over a short period of time. And CK18 is one of those biomarkers. All right, so what is CK18? Cytokeratin 18, CK18 is also sometimes referred to as keratin 18 or K18. CK18 belongs to the class of intermediate filaments, so it's part of the cytoskeleton. It's exclusively expressed in epithelial cells or cells of epithelial origin. And in the liver, CK18 is highly abundantly expressed in hepatocytes, and it actually comprises 5% of the total liver protein. Again, it's part of the cytoskeleton, so its main function is structural. CK18 is also associated with hepatocyte ballooning. Hepatocyte ballooning is one of the key histological features of MASH, and ballooning is caused by injury to the cytoskeleton, and it's been shown to be accompanied by a loss of cytoplasmic CK18 staining. CK18 is also a component of Mallory-Dink bodies. Now, during apoptosis, CK18 is cleaved by caspase, and both the full-length CK18 and caspase cleaved fragments are released into the circulation, and they can then be measured in blood samples, serum or plasma samples. Therefore, CK18 and the caspase cleaved CK18 fragments are considered biomarkers for hepatocyte death and liver injury. And there are standard immunoassays for measuring CK18 and its fragments. These assays come in two varieties. The M65 assays include the M65 ELISA and the M65 epidemic ELISA. These assays are based on two antibodies that are named M5 and M6, hence M65. And these antibodies recognize all forms of CK18, so both intact CK18 that is released from necrotic cells, as well as the CK18 fragments that are generated during apoptosis. The M65 assays therefore measure total hepatocyte cell death, both apoptosis and necrosis. In contrast, the M30 ELISAs are specific for apoptosis only. The M30 assays include the M30 apoptosens and the M30 cytodeath assays. The M30 antibody in these kits specifically recognizes a neoepitope that is exposed after CK18 is cleaved by caspases. And caspases are activated during apoptosis. And because that M30 antibody only detects those caspase cleaved fragments, they therefore only measure hepatocyte apoptosis. So again, just a quick review, the M65 assays measure total cell death, and the M30 assays measure specifically apoptosis. Both assays come in a standard ELISA format, and they can be used with serum or plasma samples. What does hepatocyte death have to do with MASH? About 20 years ago, Feldstein and colleagues observed that hepatocyte apoptosis is significantly increased in individuals with MASH, and that the extent of apoptosis correlates with disease activity. When you get excessive accumulation of fat in hepatocytes, it can cause ER stress and mitochondrial dysfunction, and this can lead to hepatocyte apoptosis. Hepatocyte apoptosis is now widely recognized as one of the hallmarks of MASH. Hepatocyte apoptosis in turn can activate immune cells and hepatic stellate cells, which then leads to chronic inflammation, liver injury, and fibrosis. So since hepatocyte apoptosis is thought to be an important, if not a critical driver in the progression of MASH, therefore, hepatocyte apoptosis is thought to be a driver in the progression of MASH. All right, and apoptosis is really a central node in MASH pathogenesis. As I mentioned before, MASH is a complex and multifactorial disease, and there are many different pathways that are thought to contribute to the disease progression. And apoptosis is downstream of most of those pathways. Shown in this slide are some MASH drug candidates and the pathways that they target. And since apoptosis is, again, a downstream effector of most of those pathways, a drug that modulates any of these pathways will ultimately also affect apoptosis. Since apoptosis is such a prominent feature of MASH, CK18 has been extensively studied as a biomarker in the context of MASLD and MASH, and specifically for monitoring the progression of MASLD. There are numerous research studies that have looked at a correlation between CK18 levels and MASH. As I mentioned, as observed by Feldstein, CK18 has been shown to be increased in both children and adults with MASH compared to healthy individuals or individuals with simple steatosis. Because apoptosis is thought to be one of the drivers in the progression of MASH, elevated CK18 levels may mean active disease. So the more CK18, the more active disease. I'll show some more data in a few more slides, but CK18 levels have been shown to correlate with the NAS and also with fibrosis. So in MASH clinical trials, monitoring CK18 levels may be useful for assessing the effect of treatment on disease activity. So that could be helpful, especially in early phase studies, because you may be able to see changes in CK18 before you see any changes in liver histology. All right, so let's dive into some of the MASH clinical trials that have used CK18 as a biomarker. And on the left, you'll see a list of all the drug treatment trials that have included one or both of the CK18 biomarkers, so either M30 and or M65. And I'll realize that this is obviously way too small to read. If you'd like to get a readable version of this table, just reach out afterwards and I'll be happy to send that to you if that's of interest. But anyhow, there are up to date over 70 trials that have used CK18. Again, these are only the MASH therapeutic intervention trials. The trials range from phase 1 all the way to phase 3. For 4 weeks all the way out to 96 weeks, and they represent drugs with many different mechanisms of action. And the point of this slide is really just to point out the wide variety of MASH trials that have included CK18 as a biomarker. And in the next slides, I'll show you some data from some of these trials. All right, so here are some examples of data from MASH trials that have used CK18 as a biomarker. On the top left, we have denifanstat, which is a FASN inhibitor, then lanifibranor, pan-PAR agonist. Bottom left is Eli Lilly's latest 3G agonist, retatrutide, and then on the bottom right is obeticholic acid. Each of these drugs has a distinct mechanism of action, But in each study, CK18 levels were significantly decreased in the drug treatment group over the placebo group. So what this illustrates is that CK18 is a drug mechanism of action agnostic biomarker. Statistically significant changes in CK18 were seen as early as 12 weeks for denifanstat. They may be seen even sooner. I mean, I've seen some data from studies that have used a direct caspase inhibitor, and you can see changes as soon as 24 hours after treatment. But again, that has to do with the mechanism of action of the drug that was used. And the point is really that the observed time course of the changes in CK18 are likely dependent on the mechanism of action of the drug candidate. As I mentioned earlier, resmetirom or Rezdiffra is the first approved drug for MASH, and CK18 was included in both the phase 2 study. That's when resmetirom was still called MGL 3196. And we're super proud that CK18 was also one of the biomarkers that was measured in the Maestro-NASH study, which was the pivotal trial that led to the FDA approval of Rezdiffra. And in both studies, treatment with resmetirom caused a dose-dependent decrease in CK18 levels as compared to placebo. All right, finally, I wanted to share some data from the Gilead and Novo Nordisk study that is looking at the combinations of semaglutide, or I call it semaglutide. It could be called semaglutide. I don't know, but anyway, I'll call it semaglutide. So it's semaglutide and firsocostat and cilofexor. Again, these are 3 drugs with very different mechanisms of action. And I thought that it was important to include these data because, one, I believe that there will be more combination trials in the future. Again, MASH, it's a multifactorial disease that may require more than one drug for treatment. But my second point for including the data is that one of the drugs in the study is semaglutide. Semaglutide is already approved for diabetes and obesity, and it will likely be the next approved drug for MASH. And unless you've been living under a rock for the last year, you'll know that the prescriptions for semaglutide have exploded in the last year. So there will be many, many patients that could be eligible for MASH trials that are on semaglutide. And what that means for drug developers is that they will have to show that their drug has an added beneficial effect on MASH in these patients that are on semaglutide. Firsocostat and cilofexor have very different mechanisms of action than semaglutide. And what you can see here is that there were greater improvements in CK18, along with some of the other biomarkers in the combination treatment arm as compared to the semaglutide monotherapy arm. And this indicates that CK18 could be a suitable biomarker for combination trials or for trials with non-native patients, so patients that are already on a drug for the treatment of MASH. All right, so I'll finish up with a few slides on the correlation between CK18 and liver histology. These data were published a while ago, but CK18 has also been around for a while. CK18 levels do correlate with liver histology, specifically with the NAS and fibrosis. For the NAS, the higher the NAS grade, the higher the CK18 level. And while CK18 is not a fibrosis marker per se, CK18 levels do also correlate with fibrosis stage. But what about changes in histology? If CK18 is to be useful as a treatment response marker in MASH clinical trials, then changes in liver histology should be reflected by changes in CK18. These are data from the PIVENS and tonic trials where patients were treated with vitamin E and then either pioglitazone or metformin. The data are from a retrospective study that looked at the relationship between longitudinal changes in CK18 levels and changes in liver histology. And what I want you to realize here is that these graphs don't show the effect of treatment on CK18 levels, but rather trial participants were grouped into histologic responders, so those that achieved resolution of MASH and those that didn't. And in each one of the graphs, the responders are shown in the open circles, and then the non-responders are in the closed circles, and change in CK18 is shown on the y-axis. And in both trials, the histologic responders had significantly greater decreases in CK18 than the histologic non-responders. But what I thought was even more interesting is that they found that early decreases in CK18, so at 16 weeks for Pivens and at 24 weeks for tonic, was significantly associated with histological changes at the end of the study, so at week 96. So in these studies, changes in CK18 were not just reflective of changes in liver histology, but they were also predictive of changes. Here's a bit more recent data from a shorter trial. This is data from the phase 2 study of selonsertib and simtuzumab from Gilead. This was a 24-week study. Again, this is not an analysis of treatment effect, but again, a correlation of changes in CK18 levels and changes in liver histology. They looked at both M30 and M65 and at changes in fibrosis and inflammation. Again, patients were divided based on the histological response. So the histological responders are shown in green, the non-responders are shown in red. And as you can see, the histological responders had greater decreases in CK18, both M30 and M65. All right, finally, and I'm really not going to go into much detail at all, but this was a retrospective analysis of data from the Sirius Eminence trial and also a meta-analysis of 27 additional MASH trials that included over 3,700 patients. And they looked at several biomarkers and their correlation with changes in various parameters of liver histology, and then they picked out the 3 best, which they found to be CK18, AST, and HbA1c. And they came up with this composite score that they termed the Z-score. In the conclusions, the authors stated that this score may be used instead of a biopsy to assess efficacy of drugs in early development. And while this Z-score may not be ready for prime time because it hasn't been validated, and it definitely hasn't been validated for individual subject assessment, it is reassuring to know that CK18 performed so well in this retrospective and meta-analysis. All right, so just to finish up, in summary, I hope that I was able to show you today that CK18 is a disease activity marker. Apoptosis and necrosis are underlying process in the progression of MASLD and MASH. If you have a lot of apoptosis and necrosis, you get a lot of CK18, you have a lot of active disease. Hepatocyte death, apoptosis is a central node in MASH progression, and that makes CK18 a drug mechanism of action agnostic biomarker. CK18 is reflective and predictive of liver histology. So CK18 levels correlate with both the NAS grade and fibrosis stage, and changes in CK18 correlate with changes in liver histology. And just some rationale for including CK18 in MASH clinical trials. I mean, first of all, it's a non-invasive biomarker. You don't need a biopsy. It uses straightforward technology. ELISAs are very widely available. It uses serum as a sample, plasma as a sample. So if you're already collecting serum or plasma samples, it's super easy to just tag on a CK18 assay. And it's relatively expensive, at least compared to some of the other technologies that are available. CK18 is an early indicator of treatment efficacy. Changes can be seen within weeks, early as 12 weeks, definitely before you may be able to see significant changes in liver histology. Also, early changes in CK18 have been associated with histological changes later on. So CK18 can support and confirm the results of other biomarkers. And in the end, what you really want are biomarkers that will convince you or maybe your investors that your drug is working and that you should be proceeding to the next phase. So what you want are arrows that are all pointing in the right direction, and CK18 can be one of those arrows. All right, so with that, I want to thank you, and I am ready for some questions.
All right, I think that's where I'm going to jump in and help out with fielding the questions. Jessica, that was a great job. It was super informative. Our first question comes in from our friends at Inspiro. Hi, Sue. So it kind of deals with using these markers for translational studies. And they said, so Inspiro has tested the M30 and M65 in their 3D liver models. And yay, they say the kits are very sensitive, and they wanted to know if you would recommend longer cultures for better windows in response to mimicking clinical trials, or very short term, few hours or days, because apoptosis happens more quickly in vitro.
That's a good question, and I am not super familiar with spheroids. I mean, that is not my wheelhouse. But I mean, so one of the advantages of CK18 as a biomarker is that you can look early. I mean, since it is a sensitive assay, but you don't have to. I mean, so CK18, if you're looking in culture serum, will accumulate in the culture serum. So, so you could look at longer time points. I mean, you don't have to catch apoptosis right as it happens, because the product of apoptosis will be in the cell supernatant. So you could look at longer time points. But yes, that would be my answer for that. But of course, if you guys want to do some studies looking at different time points, we would definitely welcome that.
Right. And also just the point that it's a great translational marker because it has clinical relevance, and also can be studied in culture systems or models, organoids, spheroids in your case. So thank you for that.
Super happy to hear that it worked for you guys.
Yeah. So also, the next question is one that we've been getting more and more. It deals with the new category of MASLD and wanting to know, can CK18 be used to study MASLD?
Yeah, I mean, yes, we have been getting that question, and I think that is super interesting. I mean, there is no data available as of yet using CK18 with MASLD. I'm actually not aware of many biomarker studies that have looked at specifically MASLD because it is such a new category. But I mean, CK18 it's not specific for MASH. I mean, other liver diseases like alcoholic liver disease, it's also a great biomarker for that. So I can see that it would be a useful biomarker for MASLD, but we just don't have any data available. But again, if anyone is studying MASLD and wants to look at CK18, please get into contact with us. We would be really interested in that. Maybe there are differences between M30 and M65 in patients with MASLD compared to patients that have pure MASLD and that have pure alcoholic liver disease. So yes, I mean, if anyone is studying that, let us know.
Or retrospectively, I mean, I think I've heard people talking at AASLD about how people will be digging into, old blocks of data to kind of harvest that information.
Yeah.
All right, so another great questions come in about essentially screening to identify subjects in the context of clinical trials that are like at risk or being able to predict disease for trial purposes or on an individual subject basis.
Yeah, I mean, there is some data that's available in that regard. I mean, we're hoping that we can generate some more data. I mean, my bias is I think it's a great biomarker for subject selection. I mean, it has been shown to correlate with ballooning. So we know that it's been used for pre-screening patient to enrich your study population for patients that have ballooning. But we don't have any validated cutoffs or anything like that. So again, my bias is I think it would be a great biomarker because when you have a lot of CK18, you have active disease, and these patients that have very active disease, maybe better responders to your treatment.
Mm.
So that would be my rationale for using it. But again, we don't have any, you know, again, validated cutoffs or anything like that. But we're hoping to get some more data in that regard in the future.
Right. And the next question kind of leads into the cutoff part. This person is wanting to know what cutoff levels were used in some of the trials that you presented, in the clinical studies that you presented, and how or who defined these levels, and were the same levels used? I know this is a really thick
Right.
question as far as
Yeah,
like
I mean,
the
no, I
nitty-gritty
think that's
details.
right. I mean, so as far as like the correlation between changes in CK18 and changes in histology, so that Pippin's, that retrospective analysis of the Pippin's and tonic trial, they actually looked at odds ratios, so different decreases in CK18 and changes in histology. And I don't know those numbers off the top of my head, but they looked, you know, at a decrease of 150 units per liter in CK18, and that had an odds ratio of whatever it was for
So more
changes
about
in
the delta
histology.
than a cutoff.
Right, and then a 250 decrease had a higher odds ratio. So again, there's nothing that's that's been validated or anything like that. I'm not sure if that answers the question, and if it doesn't, please reach out again, and maybe I can elaborate a little bit more.
Yeah, so we can probably go back and more specifically address their question afterwards via email too.
Yeah.
And then the final question that has popped up is CK18 in MASH subjects all coming from hepatocytes, or are there speculations that it's coming from a different cell type?
Yeah, that's a great question as well. I mean, so CK18 is, as I mentioned in one of the introductory slides, I mean, it is highly expressed in hepatocytes. It is also expressed in cholangiocytes, but I don't know, you know, on a mass basis what the ratios are. It is expressed in all epithelial cells. Or cells of epithelial origin, so it could be coming from other tissues. But again, in MASH patients, at least in the context of a MASH clinical trial, generally you try to rule out other diseases. I mean, you try to focus on patients that have MASH. So I would say that it mainly comes from hepatocytes.
Okay, well, that was a very engaged question and answer session. So thank you for everybody that chimed in and asked your questions. And hopefully we can send out, you know, an email that kind of more focused answers if we didn't get to the meat of what you were interested in hearing about. So with that, I think I can maybe sign off for everybody and say thank you so much for your attention. And thank you, Jessica, for a really well done, to the point presentation. Sure.
Thanks everyone for joining.
Thank you. Bye, everyone.